So two strands are ready here. One is the parent strand and this is the complementary strand. Let me introduce one more stage here. Say for example, this is one more stage in the process of replication. Now it becomes like this. One parent strand and one complementary strand, new strand. Yeah, we can say new strand. So these two intermingled DNA needs to get separated in order to move to the new daughter cells. Okay? For this intermingled DNA to get separated, there is one more enzyme which is going to act here. This enzyme separates the intermingled DNA to pass on to the next generation, and this enzyme is topoisomerase 4. So there is two topoisomerases. One is the topoisomerase 2 and topoisomerase 4. You know, difference between the topoisomerase 4 and 2 is this. It is having 3 hands. One is two are cutter hands, and one is the ligase hand. That means one introduces the cut and another reseal the things. Now you know what fluoroquinolone do. Say for example, this is the fluoroquinolone here. This is the fluoroquinolone. What fluoroquinolone do? When it enters into the body, it enters into the cell through passive diffusion. Then after that passive diffusion, it reaches to the site of action. The site of action is fluoroquinolone acts on the DNA gyrase 2 and DNA gyrase 4. Okay? Where is this actual point where it hits the DNA? It is going to inhibit the ligase end of the topoisomerase 2 and or topoisomerase 4. When it binds the, say for example, when it binds to the ligase end, that means here the hands are open, cutting hands are open when it introduces the cut. So cutting hands are open, it is going to introduce cut and cut. There is no resealing. When there is only cutting of the DNA, no resealing, it leads to the lysis of the cell. This way the fluoroquinolone acts. Okay. Now what is the preference, where it acts at the 2 and where it acts at the 4? You know, most of the gram-negative bacteria, the preference is this topoisomerase 2, and most of the gram-positive bacteria, the preference is topoisomerase 4. Okay? Where the selection, that means why humans are not affected, why human enzymes are not affected. You know, the concentration, the effective concentration in bacteria is 0.1 microgram per ml. When the concentration reaches, that fluoroquinolone concentration is 0.1. This much is not effective against the human cells, human enzymes. For human enzyme to get inhibited by the fluoroquinolone, then the concentration must reach around 100 to 1000 microgram per ml. And this is the selectivity of the actions of the fluoroquinolones against bacterial cells only. Hope you understand the things. Okay, thank you.